This is Michael Pietras, recently graduated from Tufts University, presenting aTEMPO, Pathway-Specific Temporal Anomalies for Precision Therapeutics. aTEMPO is a tool for anomaly detection in precision medicine that is heavily based on TEMPO, a previous method from our research group. TEMPO was developed to detect what we call temporal dysregulation, pathways where there is some age or time-related normal pattern of gene expression in healthy individuals that breaks down or is disrupted in unhealthy individuals that have some developmental or degenerative disorder like autism, Huntington's disease, or Alzheimer's disease. TEMPO does this by constructing a model of age as a function of gene expression for genes in a given gene set for healthy patients. Gene sets where a good model can be constructed are likely to have that normal pattern of gene expression that's related to healthy aging. Gene sets where this model fails to predict accurately on patients with a disease are therefore gene sets where we can expect the pattern to have broken down. aTEMPO works similarly, except once the age models are constructed, they're used to generate predictions for patients of an unknown class. Prediction error surprisals for a patient and gene set capture how anomalous that patient's expression-age relationship is for that gene set. We use the sum of these surprisals for all gene sets as an anomaly score for a patient. We ran aTEMPO as well as several comparator methods on 5 data sets we previously used with TEMPO. For each data set, we used a training set consisting of randomly selected 2/3 of the data marked as normal and a test set consisting of the remaining normal data and all samples with the disease. Each method computed an anomaly score for each sample in the test set, and we compute the area under the ROC curve to measure how well the anomaly scores distinguish normal samples from those with the disease. We performed this procedure 5 times, and we show AUCs averaged over the 5 replicates. Most methods perform well on the COPD dataset, where the signal is least time-related, and the BPD dataset, which covers the shortest time period. Traditional ML approaches perform poorly on other datasets, but methods which take biological relationships into account perform better. Combining temporal information with knowledge of gene sets with aTEMPO yields the best results, however. In addition, for datasets where we knew disease subtypes, we evaluated anomaly detection performance when distinguishing each subtype from normal. aTEMPO is also the top performing method here and generally performs well. The exception is on asymptomatic Huntington's, which no algorithm predicts well. However, by using only those gene sets that we find to be dysregulated with TEMPO when comparing normal patients in each replicate to the symptomatic patients, we're able to obtain far better performance even on the asymptomatic patients. This means that in domains where we have some prior knowledge about the types of anomalies that we're looking for example, early detection of a known disease, we can likely increase the predictive power of gene set-based anomaly detection methods. While this is not a particularly exciting result for Huntington's disease, where pre-symptomatic status can be very easily assessed, this does mean that if we have the data, we might be able to use aTEMPO to do early detection of more interesting diseases like Alzheimer's disease. Finally, we can also use aTEMPO as a single sample method. Since aTEMPO produces a surprisal value for each individual patient and gene set, we can use the matrix of these surprisal values to obtain information that we can use in a similar way to traditional single sample methods like GSVA, PLAGE, single sample GC. However, while we can't objectively evaluate the precision medicine applications on any of our data sets due to a lack of additional patient information, however, we were able to find some support for aTEMPO's findings via other methods. For example, in the Huntington's disease dataset, 3 patients have surprisal values 3 or more standard deviations above the mean in the positive regulation of sodium ion transmembrane transport gene set. Disruption of acid-sensing ion channels has been observed in Huntington's disease models and patient brains, and connectivity map queries for these patients suggest as treatments for these patients drugs affecting ion transport and ATPase inhibitors. So these results show that by intelligently combining temporal information with gene set information, we can greatly improve our ability to detect and characterize time-sensitive human diseases like autism, Huntington's disease, or Alzheimer's disease. Tempo and soon aTEMPO are available in an R package available on CRAN, the Tempo R package, if you are interested in checking it out. And thank you.